This video we're going to discuss a little bit about the structure of DNA, and as we think about the structure of DNA, we need to think about how does DNA and its structure allow DNA to do what we know genes do. So we know genes carry information from one generation to the next. We know that genes determine the heritable characteristics of organisms, so it determines what your eye color is and your hair color and things like that. And we know genes must be able to be replicated or copied easily so they can be in all of our cells. And so when we were talking about the structure of DNA, it needed to relate to all of these things that we know genes must be able to do. And so we'll just define DNA. DNA stands for deoxyribonucleic acid, okay, otherwise known as DNA. DNA is going to be nucleic acid made of nucleotides, of course. Nucleotides are nucleic acid monomers, joined into strands and chains by covalent bonds. So they form a chain via covalent bonds, and so of course we remember monomer and polymer. Nucleotides here are the monomer, and DNA is one of the polymers of nucleic acids. Just a little bit more about nucleotides. They are, of course, the monomers of nucleic acids made up of a 5-carbon sugar. In the case of DNA, that 5-carbon sugar is deoxyribose, hence that part of the name. And, of course, it's a sugar ending in -ose. A phosphate group, and a nitrogenous base. So we drew these in the biochem unit just to look like that. This is sort of on its side right now. So I'm going to move this to the next slide and have a better picture here. We have 4 bases of DNA. The bases are adenine, which we represent with a capital A, guanine, which we represent with a capital G, cytosine, which we represent with a capital C, and thymine, which we represent with a capital T. And here I will circle for you individual nucleotides. So again, notice that in a strand they do all basically look like the picture of nucleotides that we had drawn before, okay? And notice that these are bonded together with a covalent bond between the phosphate of one nucleotide and the sugar of another. What really changes that makes the bases different are the nitrogenous bases. And there are 2 main types of nitrogenous bases. There are basically large bases called purines and short bases called pyrimidines.